I'm going to start with where we left off last time, which is looking a little bit at neurotransmitters. And remember, you can think about this whole neurotransmitter receptor as well as this intracellular pathways as a whole system, right? And so here we can talk about this whole system of neurotransmitter receptor and intracellular pathways, right? And we can have slightly different responses for each, and there's ways to discuss these as whole systems, such as like cholinergic signaling means that we're using acetylcholine, but remember, acetylcholine can be excitatory, inhibitory, depending upon the receptor or the tissue that we're looking at, right? And so we can say cholinergic signaling at the neuromuscular junction, right? Then that hopefully jogs your memory saying, oh yeah, that acetylcholine receptor there is actually going to be excitatory, right? If we say cholinergic signaling at cardiac muscle, then you know that receptor, when the acetylcholine binds to it, is causing an inhibitory, right? So we can talk about these as systems, right? As a combination of neurotransmitter, receptor, and the intracellular pathways. Intracellular pathways are super complex. I mean, it's like a circuitry inside that cell of what goes on. And so we just don't have time to go into this. Next thing we're going to look at is some examples of neurotransmitters. You don't have to know them all. I'm just going to go through some of these that are really important that we're going to run into. Acetylcholine we've talked about already. The other ones, just so that you understand how to pronounce them: dopamine, serotonin, norepinephrine, GABA, glutamate, substance P, endorphins, adenosine, and endocannabinoids. All right, and so I'm going to have these written on the board behind the screen here, and so I'm going to talk about briefly what each one does. Some of these you may already know, some of the functions behind them. All right, so taking a look at acetylcholine. Acetylcholine, we can talk about as a major neurotransmitter involved with skeletal muscle contraction as well as smooth muscle and cardiac muscle, so muscle contraction in general. We're also going to see it's a major neurotransmitter, for communication between neurons. And so we'll talk about that one, especially when you get into the autonomic nervous system. All right. The next one is dopamine. The dopamine, is sometimes abbreviated DA, and that is generally going to be involved with kind of a, feel good, type of emotional response, but also it seems to be involved with reward. So being able to feel good when you do accomplish something. There's also some component of anticipation with dopamine. Anticipating something good happening, is also seems to be related to dopamine. Serotonin, which is sometimes known as 5-HT, you might see that written when you look up serotonin if you're interested in doing more research on it. Okay, and I could tell you guys, why it's called 5-HT if you guys are interested. Just ask me outside of class. It's basically a mood stabilizer. It's a mood stabilizer, serotonin. Turns out serotonin also is really important in aspects of gastrointestinal motility. So a lot of people don't realize one of the highest concentrations of serotonin is actually in the GI tract. All right, so the next one is norepinephrine, and its related molecule, chemical structurally related molecule epinephrine. And this is a molecule that's involved in fight or flight response. So whenever you are startled, right? You have heightened awareness. Your heartbeat increases, the rate increases. So it's a fight or flight response. Seems to also be involved with focusing, right? So fight or flight and focusing. The other two in here, GABA, and once again, this is an abbreviation, so if you guys want to know what the actual chemical name is, let me know after outside class, happy to tell you guys. This is the main inhibitory neurotransmitter in the central nervous system. All right, so GABA is the main inhibitory neurotransmitter in the central nervous system. All right. Glutamate, right, for those that maybe remember, glutamate is actually an amino acid too, all right? Glutamate is the main excitatory neurotransmitter in the central nervous system. So we have two that are kind of opposites here. GABA is the main inhibitory neurotransmitter in the central nervous system. Glutamate is the main excitatory neurotransmitter in the central nervous system, excuse me. All right, everybody with me so far? Okay. Substance P. Substance P is a molecule among other molecules, there's other ones out there, that signal pain. So it's a pain neurotransmitter. There's other ones out there as well. But substance P is one that's important for signaling pain. And the next one, endorphins, is a class of neurotransmitters that are involved with inhibiting pain signaling, including signaling coming from substance P. All right? And so there's other classes of neurotransmitters that do this. A lot of them are used to manage pain. And we'll talk about a few of those today. All right? And next one is adenosine. Adenosine does a lot of things. One is that it's important in regulating the sleep-wake cycle. It has effects on the circulatory system. And probably one of the things that's really interesting is that caffeine is antagonizes the effects of adenosine. All right, so caffeine is going to inhibit the effects of adenosine. Right. So caffeine helps you stay awake. In a lot of cases, it can help you focus. A lot of times it can have effects on the cardiovascular system. It's doing that through a lot of times affecting the same receptors that adenosine's affecting. Caffeine does a whole host of other things because it has other targets, but it mainly is, one of the main things it targets is adenosine signaling, and the effects of adenosine. All right, so last one, right? So hopefully, if you look at even the name, right, what might this be involved with? Endocannabinoids. THC, yeah, absolutely. So it turns out for a long time they didn't they found the receptor for THC, but they weren't able to find initially some of the molecules that our body makes that bind to that receptor. And so that came afterwards, that they found that these, we had these cannabinoids in our body, and whenever we talk about endogenous or endocannabinoids, those are things that generally we make or our cell makes. And so endocannabinoids are kind of THC-like molecules that our body naturally makes. All right? And so what is THC? So THC is mimicking these effects of endocannabinoids, right? And so what are some effects of endocannabinoids you might expect? So appetite, it does regulate appetite, absolutely, right? So memory, yep. So there's effects there. So you guys are hit two of the main ones. The other one is nausea. So there's some reports that, THC is able to help individuals with nausea, right? Those with, say, certain types of nausea, right? So here, endocannabinoids, they're basically molecules that our body makes that bind to the same receptor, cause the same signaling events as THC does, all right? Yeah. I think there's some literature out there on that. I'm not too familiar with it, so I can't speak to it, but it's a good question. And if people find anything out about that, let me know, if I get some time to look into it, I'll get back to y'all on that, But yeah, so, there's studies that are well done and studies that are not so well done. So that is a bit of a murky literature to wade through. So. Oh, cool. Well, I'll take a look at it, and yeah, we can take a look and see, yeah, cool. Yeah. So any questions on some of these neurotransmitters? So you guys will see some of these neurotransmitters and their functions again in the lab. All right, and so we're going to go over these guys over and over again. Okay? All right, so all of these guys, right, your body makes. So we're not talking about anything that is coming outside of our body. That's the next step, is to talk about, say, drugs, poisons, toxins, and how they interfere with some of the normal things that these neurotransmitters are supposed to do. All right. Okay, so I also have some images up here. So when you guys get a chance, all right, so see if you can match these images with some of the things we talked about, the functions we talked about for these drugs. Or not drugs, but these neurotransmitters, all right? Okay, so we're going to take a look at drugs, toxins, and neurotransmitters here. The first one, for those that had me for 231, this probably looks somewhat familiar to you. The first one we're going to look at is botulinum toxin. All right, this is made by a microorganism, and it makes this toxin. What does it end up doing? Well, it's going to interfere with these snare proteins. Remember I told you guys about how neurotransmitters get released? That you have a vesicle with a neurotransmitter and it's got some proteins sticking out from it. And the cell membrane that vesicle is going to fuse with also has some proteins, and they kind of come together when calcium enters into the synapse, right? So hopefully you guys remember that. I was telling you about snare proteins for this specific reason. Turns out botulinum toxin is going to interfere with these snare proteins, right? So proper function of snare proteins is needed for acetylcholine to be released from that presynaptic neuron. So if we get botulinum toxin that goes in and it chops up the snare proteins, right? Are we going to get these neurotransmitter vesicles to dock with the cell membrane to release their neurotransmitter? No, right? What does that mean for the postsynaptic cell, the effector here? Is that going to respond? No, it's not going to respond because there's no signaling. There's no neurotransmitter being released, right? So if we think about skeletal muscle, what's going to happen to skeletal muscle? Is it going to stay contracted or relaxed? Way relaxed, exactly, right? Because we need acetylcholine in order to initiate a contraction response. We don't get acetylcholine released, we're not going to initiate a contraction response, right? It's a big problem. Okay? And so that, when we stay relaxed, we say that's flaccid paralysis. Okay? So when the muscle, skeletal muscle stays relaxed, doesn't contract, we say that's flaccid paralysis. Any questions before we move on? They're little proteins hanging off of vesicles and also embedded in the membrane. And what they end up doing is they twist each other together when calcium's present to help pull the vesicle into the cell membrane. And when the vesicle gets to the cell membrane, those fuse and they create a little opening that allows release of the neurotransmitter. So if you don't have proper functioning of snare proteins, what ends up happening is you don't get this fusion event so that this opening is generated so that neurotransmitters can be released. Yeah, thanks for asking. Any other questions before we move on to the next? All right, another one, another toxin, tetanus toxin, and also a toxin generated by a microorganism. Also affects snare proteins, believe it or not, but not on the motor neuron. It affects a neuron that regulates the motor neuron. Okay? So if we have skeletal muscle and we have our motor neuron that talks to our skeletal muscle, we're talking about a neuron that talks to the motor neuron. Okay? All right? Everybody with me so far? So in the normal situation here, here's our motor neuron. We get our action potential, and then we get acetylcholine release that binds to the acetylcholine receptor on the skeletal muscle. Everybody with me on that? All right, so here's our motor neuron. Okay? Here's our skeletal muscle down here. Here's the receptors on the skeletal muscle. So if we have normal functioning motor neuron, we have an action potential that comes down here, causes acetylcholine to be released, right? Binds to the acetylcholine receptor on skeletal muscle, causes contraction, right? Okay, everybody with me so far? Those are normal events for skeletal muscle contraction. Turns out, in order for not too much acetylcholine to be released, there's a bit of a neuron that puts the brakes on that motor neuron, right? And so that's this neuron here that releases this inhibitory neurotransmitter that makes sure that we don't release too much acetylcholine. All right, everybody with me so far? So so here we haven't talked about this situation before, but basically the whole idea is that the amount of acetylcholine that gets released is somewhat regulated by another neuron, right, that's interacting with the motor neuron. Everybody with me so far on that? Okay. Right. So here, this inhibitory neurotransmitter is something called glycine. This is a main inhibitory neurotransmitter in the spinal cord. Okay? And so this is going to be making sure we don't get too much acetylcholine released. What this toxin does, all right, is that it blocks this. So glycine is a neurotransmitter. In order to get release, it too needs proper functioning of snare proteins up here. So the botulinum toxin, not botulinum, the tetanus toxin here, all right, interferes with this. So if you take the brakes off, right, what ends up happening? Do we get more or less acetylcholine released? More, right? So we get more than we should, which what happens to the skeletal muscle in that case? So is it going to be staying more contracted or more relaxed? More contracted, right? Okay, that's exactly what happens. All right, so we're just kind of moving upstream, further away from the skeletal muscle where the site of action is for this toxin. Okay, all right, so it inhibits glycine release. So glycine is not allowed to inhibit acetylcholine release, which means the skeletal muscle stays contracted, and they don't get this relaxed signal, and so here we can get things like lockjaw. This type of paralysis, when we stay contracted and not relaxed, is called spastic paralysis. All right? So the two types of paralysis, flaccid and spastic. Flaccid, meaning that we're relaxed. Spastic, meaning we're staying contracted here. Right? And so this is the scenario we're talking about. We've blocked our ability to release glycine, which means now we're releasing more acetylcholine than we did before, right? Which is causing our muscle, skeletal muscle, to stay contracted longer. Okay?
Is this new protein on glycine itself, or is it on the...
It's on the, because this is a neurotransmitter,
Right.
and glycine is in the vesicles, right? So in order for this neurotransmitter to get released, it has to have proper snare protein function.
And the main function of snap protein is just to pull it closer to the membrane?
Pull it closer to the membrane and then allow it opening for the neurotransmitters to get released. So here we're talking about where's the toxin site of effects, or which neuron are they working on. Here, this tetanus toxin's working on this inhibitory neuron. Previously, the botulinum toxin was working directly on the motor neuron. Yep. So we're just saying, you know, there's some specificity, which is actually super cool with toxins. You know, how does the botulinum toxin, you know, affect the motor neuron versus the tetanus toxin affecting the snare proteins and the inhibitory neuron? It's really fascinating research. Fortunately, we don't have time to get into, right? But basically, they're affecting snare proteins just in different cells. Everybody with me on that? All right? Okay. All right, so many drugs, toxins, poisons can impact neuronal communication. They can be psychoactive, but don't have to be. And a lot of times they're influencing action potentials or whether an action potential fires. So that means that we're either making it easier or more difficult to fire an action potential. So they're basically causing that resting membrane potential to get closer to the threshold or further away. That's a lot of times what they're doing. And they do that by affecting neurotransmitter release in many cases, or neurotransmitter binding, or the amount of neurotransmitter in the synapse. All right? And so they influence a lot of those events. They influence the presynaptic neuron sometimes. They'll influence the neurotransmitter release like we just saw. They'll affect whether the neurotransmitter is able to successfully bind to the receptor, right? Or in some cases, these drugs will mimic the act of binding to the receptor to stimulate the receptor independent of the neurotransmitter. All right. So some terminology here. So an agonist binds and stimulates a receptor. So it has same effect as a neurotransmitter. In some cases, it can stay bound to that receptor longer than a neurotransmitter, meaning that it can cause the extended effects of the neurotransmitter. It can affect that receptor longer, and therefore it can have a greater effect than the neurotransmitter does alone, right? So it mimics the effects of the neurotransmitter or the ligand for that receptor. The other term is something called an antagonist. Antagonists block the receptor. So it's like someone parking in your parking place, right? So if someone's parked in the parking place, then the neurotransmitter itself can't bind, and therefore, are you going to get a response? No, right? So here, antagonists, right, inhibit or block, right, the ability of the neurotransmitter to bind the receptor, which means that you don't get a response or that response is inhibited. Okay? So agonists and antagonists. Right? So here, agonist alone full activation of the receptor. We mimic the effects of whatever that ligand is, that neurotransmitter in this case. Here, if we have an antagonist, that's going to bind to the receptor, which means that we're not going to get the ability of the normal ligand, the neurotransmitter, to bind, right? Okay. So if we had an acetylcholine receptor blocker, even though we get acetylcholine released, would we get skeletal muscle contraction if we had a perfect antagonist? No, right? Okay, does that make sense? Everybody with me on that? All right. Okay, so you can do some competition between the endogenous ligand, the antagonist and agonist, and so forth. We're not going to go into that detail, but I want you guys to be familiar with the terms agonist and antagonist, because I'm going to be using them to talk about the drugs coming up. All right? Okay. Just getting you guys to have the right perspective here. So one of the things is, where in normal flow of axon terminal events does a disruption have? Right? So if we're talking about a particular drug, is it affecting the receptor? Is it affecting neurotransmitter release? Is it affecting neurotransmitter being disposed of in the synapse? You know, is it affecting reuptake, for instance, recycling? Is it affecting the ability for it to be chemically broken down? So that's one of the things you want to think about. What's the consequences of that? Do you get more or less neurotransmitter? And then if you have more or less neurotransmitter, or if you get signaling from that receptor that's elevated or decreased, what are the effects on that cell or tissue, you think? Right? So these are the sequence of events that we need to be talking about. All right? And so here, I've listed a few of the things that we talked about earlier with neurotransmitters, including serotonin, GABA, dopamine, and norepinephrine, epinephrine. All right? Okay? And the neurotransmission lab that you guys are doing this week on Wednesday is going to be going through these functions again of these neurotransmitters. And the neurotransmission activity that I got planned is also going to be looking at the effects of drugs, toxins, and poisons, all right, on these neurotransmission events. Okay? So you guys are going to see this again. All right? So why don't we do this? Why don't we take a 5-minute break? Yeah, I did see some. Okay, so I think we'll continue our discussion here. So let's just start off with making sure you guys remember antagonist and agonist. So which one is mimicking the neurotransmitter or the ligand? Agonist, right? Which one's blocking or inhibiting the actions? Antagonist, right? All right, good, because we're going to be using these terms, at least I am, and hopefully you guys understand what I'm saying to you. All right? Okay. So one poison out there, sarin. So chemical weapons, a lot of them can affect the neuromuscular junction or events at the nervous tissue, and it inhibits acetylcholinesterase. Right? So it inhibits acetylcholinesterase. So follow that sequence of events all the way to what's happening at the skeletal muscle. So starting with inhibiting the acetylcholinesterase. What's the effect of that going to be? Let's start off with the effect on the neurotransmitter levels. So what neurotransmitter are we looking at here? Acetylcholine. What's it doing to the levels of acetylcholine? It's going up. All right, good. What do you mean? It is specifically taking out what? Acetylcholine. Yeah, exactly. So it's taking out acetylcholine, but we blocked that ability, and so here we're affecting acetylcholine levels. And what are we doing to acetylcholine levels? Raising them or lowering them? Raising them. And so what's that consequence on muscle? So what's happening to the skeletal muscle? Let's back that up a bit. Is contracting it, right? Everybody with me so far? So these are the events that you want to go through. Start with what you know, and get to the effector. So here we're causing skeletal muscle contraction, right? And we're not necessarily choking to death with esophagus, but
you're not breathing.
Breathing, yeah, absolutely. So the diaphragm's a skeletal muscle, right? And so here we have a lot of acetylcholine, which is causing contraction. What type of paralysis is that? Anybody remember? Spastic paralysis. Spastic and flaccid. Flaccid's relaxing. Spastic is going to be contracted, right? All right. Everybody following me on that? Okay, that's what ends up happening with acetylcholine esterase inhibitors. There's other ones out there. Serin's only one example. All right, so you probably have heard of some of these. How many folks have seen some of these? Right, so Prozac, Sterzone, Zoloft, Cymbalta, Luvox, Lexapro, Paxil, right? So a lot of these, they're inhibiting serotonin reuptake. So we say that they're selective serotonin reuptake inhibitors, SSRIs. For those in the healthcare field, you generally don't want to use this, because it gets confused with another acronym, all right? But SSRIs are just commonly referred to, these are referred to as SSRIs. They're inhibiting serotonin reuptake, which means what are we doing to the amount of serotonin? Elevating it or decreasing it? It's going up, right? And then what is serotonin normally involved with? Mood stabilization, right? So we're elevating or impacting moods, positively mood stabilization, right? So we haven't we'll talk about it when we get to the brain. There's a part of the brain that's called the limbic system, which is important in emotional response, emotional attachment. So when you guys are walking around and you get a visceral emotional response to something, like you walk into a room and there's a smell you don't like, that's generally triggering some part of the limbic system, right? So it's that visceral, emotional response attached to certain events. So is that everybody feeling comfortable with starting off with what we're talking about? Right, what knowledge you know, and then figuring out what the impact is on the neurotransmitter levels, and then what that impact of those neurotransmitter levels have on, right, the effector. Okay. So here, this is what we're talking about. So here's our presynaptic neuron, postsynaptic neuron. So we get serotonin being released here, and then it can be taken back up, right? But if we have a blocker, a selective serotonin reuptake inhibitor, right, that serotonin doesn't get taken back up, which means that it stays elevated in the synapse. And it is going to continue stimulating the effector cell. All right. All right, so another one, cocaine. So this one is blocking the reuptake. Primarily it has its effects on dopamine, but it can also affect other catecholamines, including norepinephrine here. All right, which means what's happening to the levels of these? We're inhibiting reuptake. Going up or down? Should go up, right? So all the things that dopamine, norepinephrine, epinephrine do, are going to be accentuated, right? Okay. And so here, stimulating pleasure center of the brain, increasing euphoria. Dopamine, remember, is involved in the reward centers of the brain, right? Emotional centers. Blocking of norepinephrine reuptake can impact on the heart. Okay. So when we get to cardiovascular section, I'll have a public service announcement about beta blockers, all right? And so we'll talk about cardiovascular events with respect to beta blockers. And so hopefully, what if norepinephrine and epinephrine are major neurotransmitters in something we call the sympathetic nervous system, okay, which has effect on heart rate and contractility. So it increases heart rate and contractility. What are these inhibitors going to do? What's cocaine going to do? what's going to do to heart rate? It's going to go up or down? Go up, right? And the force with which it contracts also should what? Increase or decrease? Increase, right? So are we elevating the stress we put on our heart? Yeah, exactly. All right. So another toxin here. So alpha bungarotoxin acts as an antagonist for acetylcholine receptors at the neuromuscular junction, right? So remember, antagonists at the neuromuscular of the acetylcholine receptor at the neuromuscular junction. What's the effect of this on skeletal muscle tissue? So it's an antagonist. Flaccid paralysis, right? So we're blocking the ability to get contraction, right? Now, if it was an agonist for the acetylcholine receptor, right, what type of paralysis would we have there? Spastic paralysis. For those that are in 231, anatoxin A was an example of that, that we did in the muscle contraction activity for extra credit, right? Was spastic. All right. So agonists of inhibitory responses. So think about it this way. Excitatory neurotransmitters are like the gas. Inhibitory neurotransmitters are like the brakes. So here what we're doing is we're pressing the brakes even further on these systems. So when we have agonists for inhibitory neurotransmitters, right, we're putting the brakes even further on the system that it controls. So here, GABA, which is gamma-aminobutyric acid, you don't have to know that. GABA is perfectly fine. So if you write in on a GABA on a quiz or an exam, I'm perfectly fine with that. All right? Has inhibitory responses in the CNS. So a lot of things are going to be affecting this system. Alcohol, BZDs, benzodiazepines. An example includes Valium. They have similar effects acting as GABA agonists, right? So if they're acting as agonists, then what does that mean? Are they mimicking the effects of GABA, or are they inhibiting that system? They're mimicking it, right? Okay. So in areas of emotion, fear, and memory, right? So those get brakes put on them. So Rohypnol. Mm-hmm. Yeah. So Rohypnol, sometimes known as the date rape drug, okay? Roofies, they affect this system. Okay. All right. So remember, when we talk about antagonists and agonists, not only can we talk about antagonists and agonists for excitatory neurotransmitters, but also inhibitory neurotransmitters. Mm-hmm. Yep. Now, in some cases, some of these drugs or toxins, they can have effects on multiple systems. And one is methamphetamines, which is going to stimulate release of dopamine from the axon terminus, and then it inhibits also the dopamine reuptake, right? So think about what's the consequence of that with respect to dopamine levels. And goes up, right? And all the things that dopamine is important for, reward, also increases euphoria, and so forth. Also affecting norepinephrine. Remember that was an epinephrine, right? That was main sympathetic nervous system. So what's that doing to the heart? Epinephrine and norepinephrine, heart rate goes up. Heart contraction strength goes up, right? So once again, we're increasing stress on the heart. Coupled with the dopamine response, with euphoria, it's quite addictive, right? So you can have these problems where individuals have these other effects on important parts of our body, like the heart, right, just to feel good. All right, so here, what can end up happening is that you can get rapid downregulation of the dopamine receptors. What do I mean by that? It means that they're stimulated so much that if you start off with 100 dopamine receptors and we added a whole bunch of dopamine in there, that postsynaptic cell is going to start trimming back the number of dopamine receptors, right? So instead of, say, having 100 like we normally would, say we get down to 50, right? Okay, everybody with me so far? Now, what if someone takes away these drugs, takes away methamphetamines? So that postsynaptic cell, instead of having 100 receptors, only has 50 now. So that response of that postsynaptic cell going to be at the normal level or less, do you think? Less, right? So these are some of the reasons for withdrawal, right? So how do we get back to feeling good? Well, we need more dopamine, right? Take more methamphetamines. So this is the cyclical problem, downward cycle. Okay, yeah, go ahead, Amanda.
I know that like people who are using that off of it, they eat more sugar.
Hmm.
So do you know what sugar releases? Is it releasing some of these like dopamine?
Could be. I'm not too familiar with the signaling pathway on that, you know. There may be some signaling pathways from that sweet receptor, you know, that are blunted. And so in order to get that, or there may be some sort of pleasure pathway cycled to it, you know. It's a good question. Another thing I'll look into when I have a chance. But no, really good questions. Yeah, go ahead. Sorry. Yeah.
Can you ever get something bad?
Yes, you can, but it takes time. And so you got to suffer diminished dopamine effects until you get back to those levels. Yeah. Yes. Yeah, you're not stimulating it as much. So what you do is, okay, we went from 100 to 50, let's say. And methamphetamines, let's say that is what we call a full agonist. So it's giving the maximum response. Methadone is maybe a partial agonist. Okay, so rather than stimulating, a whole bunch of dopamine, you stimulate a little bit. So rather than going from 100 to 50, maybe you go from 100 to, say, 75, right? And so you basically trim down back to normal levels, for instance, right? Or you go back to normal levels. Sorry, going the wrong way, right? Okay, but that's the whole idea behind not going off cold turkey, right? Is to taper people off. And so we'll see that. That's one of the public service things I'll talk about with beta blockers. It's really important. Yeah, sorry, go ahead. Oh, we're going to get to that. Yeah. Oh, yeah. Oh, yeah. Yeah. Hold on. All right. Yeah, we're getting there. Because I put a whole section on opioids because it's such a crisis. All right? All right. Intense dopamine release, especially in the rewards pathways in the brain. Right? So there's other effects of these drugs on other parts of our body which are pretty menacing. Affecting the heart, that's pretty damaging. Heart's pretty important. It's got to function properly, right? But people don't want to feel bad either. And so they continue taking, these drugs in order to feel good, but they don't realize the effects that they're having on other parts of their body, which can be pretty hazardous. Okay? So here, I've listed some of these. They can affect sleep-wake cycles. The reward centers of the brain get really elevated, highly active, for individuals taking, especially cocaine. I've seen some of the data when I was doing some research with people that are looking at cocaine, and those parts of the brain do light up, like a Christmas tree. They get very, very stimulated. All right, we're getting to, I forget your name? Cody. right? So you're talking about heroin, okay? We're going to get into opioids. Heroin affects opioids, okay? So I'm going to show you some statistics, and I'm going to show you a story that just came out today on this, all right? So here you can take a look at opioids. Heroin's one of them. Acts as a agonist of an opioid receptor. These are that mean is it stimulating or blocking the receptor? Stimulating, yep. And so here's commonly prescribed opioids, semi and natural. Here's synthetic opioids, right? Here's any opioid. And what we're looking at here is deaths per 100,000. So it's gone up exponentially here, right, in the last couple of years. It's pretty scary.
Is there any opioid line? Is that all?
That's everything together. But you can see what's driving this. The synthetics and commonly prescribed, and also, or I should say more heroin and synthetics, right, are really driving this, okay? It's pretty scary. Let me see if I can find. So this is the title of a story that just came out today. Odds of dying from accidental opioid overdose in the U.S. surpasses those of dying in a car accident. It's phenomenal. It's pretty scary. So there's a good chance, I know my family's been affected by opioid-related, issues, okay? There's a good chance that if you work in the healthcare field, or if you're living in the U.S., that your family or somebody who's affected by opioid abuse. So I want to take some time to talk about that. And I thought I saw a hand come up. Yeah, go ahead, Cody. Mm-hmm. Oh my gosh. Mm-hmm. Oh wow. It's and that's the other thing is that you cut in other things in here, so it's not pure. Mm-hmm. And we're going to see the potency, difference in just a second, which is scary. The other problem is opioid-related issues in pregnancy. So that's gone up, too. And these poor, newborns are going through withdrawal. All right? And so here is called neonatal abstinence syndrome, it's called. And that's gone up as well. Yeah, go ahead.
For the Narcan, are you going to go over how Narcan works?
A little bit, yeah. The addictability, that's a word, of opioids. So here's some data on probability of still continuing to use opioids after 1 year or 3 years, depending upon number of doses you got initially. So notice here that if you take a look at, 25% people are still using after a year, you just need 10 days of an opioid prescribed to you, right? Be able to use, almost 25%. So if you have 30 days, people are still using opioid. 30% are still using opioid 3 years after if you got a prescription that lasted 30 days, which is pretty addictive. It goes up sharply here. So you see this sharp increase. So it becomes quickly addictive even a year after, is the point of this graph. So I just want to talk briefly about opioids. So opioids are important in blunting. So endorphins, enkephalins, dynorphins. So if you look at, some of the neurotransmitter classes, endorphins block substance P signaling among other pain receptors or pain receptor signaling. So here, in addition to analgesic effects, blocking pain, right? Opioid receptor receptor stimulation has effects on mood, anxiety, and affect. How you are, feeling. Positive, are you feeling down, and so forth, right? You don't have to know all of this, but I want to point it out because it's such an issue. So we have 3 separate opioid receptors. Most of the drugs are targeting this one, which is involved with euphoria and stress coping. So in addition to pain relief, right, we're also stimulating euphoria and be able to cope with stress. So if someone comes off these drugs, right, they're not going to be able to cope with stress as well. You have, less euphoria, so they want to get back on the drugs. Okay, right? So this is a big issue. All right, so there's effects on what we call hedonism and mood, in addition to the pain relief effects. All right, is the point I want to make here. Now this is the one that Cody was getting at. So here, right, so morphine, okay, so morphine is going to be kind of common opioid given, and then everything is kind of normalized to that. So you can see fentanyl is about 100 times, right, as potent as morphine. But it doesn't stop there. There's other synthetics that are even worse. So if they had to resize the scale. So here's our fentanyl. We resized the scale so the fentanyl's here now. Look at these other synthetics that are out there, right? This guy's a tranquilizer for elephants. Look how much more potent it is, right, than fentanyl. And look how potent fentanyl is compared to, say, morphine. It's ridiculous. You need just a little bit of carfentanil, right? So there's synthetics out there that are hugely potent, all right? Okay. So you guys were asking about, some of the treatment options, right? So what I have here is I took this table out of this article. The pluses mean that they're agonists, minuses mean they're antagonists. All right, so here morphine and some of the other ones. Methadone is also an agonist, but it's not a full agonist, it's a partial agonist. So if you want to wean somebody off of opioids, you can use methadone. All right, the one that is probably not used enough is this antagonist naloxone. Right, so here we're talking about blocking the receptor. Right, so blocking the receptor. Okay, so I know we're kind of getting towards the end here, but I wanted to show you guys these tables. You don't have to know all of the drugs that we have. I have a neurotransmitter table up on Blackboard that you guys need to know. What you need to know is acetylcholine, glutamate, and substance P are excitatory, okay? And the acetylcholine, GABA, glycine, adenosine can be inhibitory. All right. You don't have to know all of which ones are direct versus indirect. The one you have to know that's direct is acetylcholine, and that it also is indirect. So where is it direct at? Neuromuscular junction, and indirect at cardiac muscle, right? GPCRs. The other one is going to be norepinephrine, also is going to be working on a GPCR. Okay? It'd probably be a good idea. All right, so this is the list. It's the same list that you guys have in the neurotransmitter table, right? These are the ones you need to know. Caffeine, alcohol, SSRIs, cocaine, methamphetamine, morphine. You don't need to know, but maybe a good idea to know fentanyl and heroin, are also opioid agonists, and naloxone is an opioid antagonist. All right, so you don't need to know all the neurotransmitters. These are the ones that you do need to look at. So, all right. Okay, so I know we went a little bit over. I apologize for that. All right, but I wanted to make sure to get through and talk about opioids in detail. Okay. All right, so we'll start in on looking at the spinal cord gross anatomy on Wednesday. And don't forget, we'll start lab at 10, on Wednesday, and we'll also do a neurotransmission activity in lab.